Our last speaker is Arun Dayanandan from the Brown Lab, and he's going to be telling us about the Addams family. So take it away.
All right, so I'll be telling you about inheriting fear, maternal background predation risk, and neophobic responses in convict cichlids. However, before I start, just with a quick show of hands, who here in one way, shape, or form thanked their mother yesterday? It's Mother's Day. There's a few. So the ones that did, I'll teach you another reason why.
Well, in Canada,
why you should thank them.
Many countries is on Mother's Day.
Well, you should be thanking her every day anyway. For those that didn't, shame on you. Hopefully after this presentation, you'll give her a call.
My mom's dead.
Dark fast. Hey, it fits the topic. All right, so first I'll start with stress. So stress is defined as a state of threatened homeostasis that is reestablished by a complex suite of adaptive responses. It's an adaptive mechanism that allows fish, in this case, to cope with real or perceived stressors in order to maintain its normal or homeostatic state. There are many causes of stress, I'm sure all of us have known, including the noise outside. So food restriction, if you're getting hangry, especially at this time of day, don't worry, we're almost done. Presence of conspecifics, if you're taking a bus or a metro to get here today, I'm sure you know how stressful it is to have your face in someone else's armpit. And then predation risk, again, real or perceived. And these can fall under four major categories: visual, auditory, mechanosensory, and chemosensory. The effects of stress itself can be divided into three major categories. Primary, so the effects of stress on hormones such as corticosteroids and catecholamines. Catecholamines being the fight or flight hormones such as epinephrine and adrenaline. Corticosteroids being cortisol, cortisone. There can be secondary effects of stress, so those on metabolism, cardiovascular function, these are systemic changes. And tertiary effects, so this is on whole body changes such as growth and behavior. So we know that stress is it's a complex suite of adaptive responses. It can be real or perceived stressors. However, it may not be adaptive in all cases. If the intensity of the stressor is overly severe or long-lasting, this effect can actually be maladaptive. And that's where mother comes in. So maternal effects are the influence of a mother's phenotype on her offspring's phenotype. This can be changes in regulatory mechanisms, effects on eggs, so hormone concentrations, it can be on embryogenesis and the development of the offspring, life history changes, offspring performance, so in particular behavior, things such as aggression, defense, and birth swimming, as well as the offspring physiology. So, generally, it was thought up until 2013 that maternal effects of stress are predominantly deleterious. These are negative things for the offspring's fitness. However, there's now a lot more evidence showing that this may not be the case. In fact, the mothers may be conferring an adaptive advantage to the offspring when they are in these periods of elevated stress. And this is done through buffering strategies. So buffering strategies are ways to protect offspring from deleterious effects of maternal stress that would otherwise compromise the progeny during highly vulnerable stages of development. This is done through two major hormones, so corticosteroids in general. However, unlike mammals such as you and I, where cortisol and cortisone are both active in us and they lead to the same physiological reactions, in fish, cortisol is the active hormone and cortisone is the inactive hormone. And this is important to understand because the mother's internal environment can actually change the cortisol to the inactive form cortisone. And we see that in the vitellogenic follicles. So for example, yolk proteins, vitamins, lipids, maternal transcripts, these are all being put into the egg during the formation of the eggs in the mother's ovary in fish. However, cortisol is able to move between the developing membrane. And so the mother's environment can actually convert this cortisol to cortisone and thus buffer at least those primary effects. So we know that there's these effects in the primary. Now what remains to be seen, and what's a large area of study now, is what are these effects on the secondary and tertiary effects of stress. In particular here, I'll be talking about behavior. So we're back to causes of stress, restriction, food restriction, conspecifics, And here I would also like to highlight predation risk once more, real or perceived. And I'm interested in looking at the chemosensory aspect of predation risk. So, you know, what smells like a predator in my environment? And from here now I'll jump to neophobia. So neophobia is defined as a generalized avoidance response to novel stimuli. It is an adaptive anti-predator strategy for vulnerable prey dealing with spatial and temporal variation in predation risk. So it's an adaptive anti-predator strategy. And so the objective of my research is to see if early stage fish spawned by mothers exposed to elevated background predation risk do exhibit neophobia. I'll be doing this by taking male and female convict cichlids, I have a female right here, and pairing them up for about 3 days until they form these breeding pairs. Once these fish have formed these breeding pairs, I'll be stressing them with 20 milliliters of alarm cue. That's a extract made from the skin of their conspecifics or siblings. And the idea behind this is that if you smell your siblings' guts floating around, chances are whatever just ate your sibling is also interested in eating you. After the, and as well, I'll also be giving the control, the fish designated as control, tank water. This will be happening 3 times per day until spawning occurs. If no spawning occurs after 1 week, then I'll switch out the pairs because, like humans, fish can be picky in choosing their mates and they don't always breed. After spawning, males and females will be left with the eggs until the larval stage, just to make sure the eggs can reach the to reach larval stage because they actually they need the parents present to hatch. However, once the larvae are present, the males and females will then be removed from these tanks and tested for neophobic responses. Then once the larvae, actually I have a picture of the larvae, so at this point is where the parents will be removed from the tank. And then once these larvae reach the free swimming stage, then they will be tested in pairs for these same neophobic responses. So fish will be tested in pairs, male and female breeding pairs, or as 2 randomly assigned free swimming juveniles. And to do this, they'll be moved to new tanks and left to acclimate for 45 minutes. At this point, they'll have a 5-minute pre-exposure recording, so I'll be sitting there measuring the time spent moving in seconds, as well as the number of foraging attempts. The number of foraging attempts is defined as the number of times a fish turns 45 degrees in the water and makes an effort to forage or pick at food in the substrate. At this point, they'll be given 10 milliliters of either a novel odor, so something they would never encounter in the wild. This can be either lemon oil or goldfish odor, or controlled, in this case, distilled water. After this exposure, there'll be another 5 minutes of recordings. So once again, time spent moving in seconds, and the frequency of foraging attempts of a focal fish. So again, the reason I'm testing them in pairs is because previous research has shown that if you do test them individually, they tend not to move at all. So in order to get a more realistic and biologically significant measurement, or set of measurements, they need to be tested in pairs. But I'll be doing one of the 2 because I don't have an extra head yet. And so a reduction in time moving and frequency of foraging attempts has been shown to be consistent with an increase in predator avoidance behavior in juvenile convict cichlids. And this is something that I'll be seeing because, as you saw earlier, if they are showing these behaviors, this means that they are indeed exhibiting neophobia, and the effects on those primary, secondary, and tertiary effects of stress are what we can draw inferences on. And so with that, I'd like to thank everyone, thank my wonderful lab, great supervisor, and the Adams family, as well as all these funding agencies and my parents.